Good morning, everybody. It's Beth Spooner from the MRC, from the HIV Infectious Diseases and Other Infectious Diseases Unit, HIDRU. We'll just wait a few minutes for more people to join. I hear some keyboards ticking, so just to remind you to mute your lines, please.
Please, just 1 second. Richard is having problems to log in. Let me just connect him.
While people are logging in, let me just do a bit of an introduction and an overview before our speakers. So, as I said, I'm Elizabeth Spooner, Beth, the Deputy Director at HIDRU, and we are hosting the symposium today. As you can see, our HIDRU is our new name as of the 1st of February. We were previously the HIV Prevention Research Unit, but with COVID and TB and the other diseases we're now covering, we renamed ourselves. So today's symposium is the interaction between COVID-19, HIV and TB with the effect of viral evolution, disease progression, and health service delivery. So that's the program that you would have seen if you're on this symposium. And so we've got 3 speakers that will speak for 20 minutes and then 10 minutes of questions and discussion afterwards. And we'll have a little stretch break in the middle if we're running on time. Just to remind you, our speakers are Richard and Kogi and Kobus, all based here in Durban, KZN. And so we really look forward to their presentations. From our side, we have chairs of the 3 talks. So there's Nobubelo Ngandu, who's in our unit in Cape Town, Marion Loveday, who's here in Durban, and Terusha Chetty, also here in Durban. And so the aim of our symposium is to present emerging insights about viral evolution, disease progression among individuals co-infected with SARS-CoV-2, HIV-1 and or TB, and the impact of COVID-19 on our health systems. So I like pictures, so a big overview, and just to remind us of our vaccination status in the world. As we know, Africa is very unvaccinated, and including South Africa. And just to also remind us that people are still dying of COVID-19 around the world. And even though we're not in an official wave in South Africa, we're still having daily deaths. And our very MRC excess death graph, as we can see, it's still hovering above the upper prediction bound. So we're still seeing excess deaths in South Africa from COVID-19, knowing that we've reported 300,000 excess deaths in South Africa over the COVID-19 epidemic, with about a third of that official COVID deaths. And the Lancet report last week showed that actually that proportion is quite international, in that many countries have a similar ratio of deaths to excess deaths. So yeah, it's a problem all around, including in America and Russia and Mexico. And then just to HIV, we know that we've been the epicenter of the HIV epidemic, and there in 2019 we see our cases being the highest of new cases in HIV. Again, also HIV and AIDS deaths in 2019 were very high in sub-Saharan Africa and South Africa. And then our co-infection rate of TB patients who tested positive for HIV. This was from 2016, but also shows the co-infection rate that we know and have come to expect in our country. And so these three diseases really are a real burden on our health system. And so we're going to look at them from the actual pathogen point of view, from the patient point of view, and from the health system point of view, through our three speakers and experts today, and to get a better understanding of how we navigate the health system in our country, and the patients, and the emerging variants that come our way. So there's the program again for everybody to have a look at. So I'm going to hand over now to Nobu, who's going to chair our first session. For Richard, we are running a little ahead of time, so we can wait for more. I see more people are joining. I can't see the screen. Ari, are we recording?
We are.
Okay, so just to say, yes, go.
There is. Right.
I am here, yes.
Ah, wonderful. Hi, Richard. Welcome.
Sorry, we were all in another meeting, Kogi, Kobus, and I.
Oh, no problem.
We were having a meeting to ourselves.
So sorry about that. Thank you.
Great. So I'm going to hand over to Nobu to chair the session. Thank you.
Thank you. So we can go ahead, right?
Yeah, please.
Okay. Good morning, everyone, and welcome to our symposium. Our first speaker is Dr. Richard Lessells, who is an infectious diseases specialist at KwaZulu-Natal Research and Innovation Sequencing Platform, KRISP, at the School of Laboratory Medicine and Medical Sciences, and he's an honorary research associate at CAPRISA. In the last 2 years, he has been heavily involved in COVID-19 work, in particular KRISP's genomic surveillance activities within the Network for Genomic Surveillance South Africa, and the broader activities in the National SARS-CoV-2 Variants Research Consortium. He is a current member of the Ministerial Advisory Committee on COVID-19 vaccines here in South Africa. So I'll hand over to you, Dr. Lessells. Just to remind you, he's going to speak on the interactions between COVID-19, HIV, and TB, zooming in on the effect on viral evolution. Thank you.
Thank you very much, Nobu. Thanks for the kind introduction, and good morning, everyone. If somebody could just confirm that you're seeing the slides okay in full screen.
Yes, we can see the slides.
Okay, great. So thanks. It's a really nice symposium that's been put together, and it's a real pleasure to be sharing the virtual stage with friends, Kogi and Kobus, and I plan to just start off really talking a little bit about the interactions between COVID and HIV from the virus evolution perspective. And what I hope to do in this short talk is convince you that there are some differences in how this virus behaves in people who have advanced uncontrolled HIV. And from a couple of cases that we've reported here in South Africa to show you that sometimes this has consequences in terms of chronic infection and intrahost evolution in extreme cases where people can't clear the virus at all. And then try and put to you that this is one possible mechanism how variants, and particularly kind of variants of concern that have specific properties like immune evasion, how these variants may emerge into the population. And then finish by just arguing that because of these things, I think we do need to take this interaction between COVID and HIV seriously, and we do need to think about it in our public health response. So let's just start by talking about the virus dynamics. And what I'm going to show you is just data from a few different South African studies that have looked at the virus dynamics in people with COVID and have some data on how that's affected by HIV and by the different parameters, the viral load and CD4 count. So the first study that I'm showing you here is from KZN. It's from the Commit KZN Cohort study. So that's the study led by Alex Segal and his team at ARI, but involves many other groups in KZN. And they enroll primarily hospitalized cases of COVID, and then they follow them up over several months with sampling at regular intervals to look for the immune response as well as the virus dynamics. And here they're just showing data from the publication last year in eLife. And here they're looking at the time to SARS-CoV-2 clearance, so the time to PCR negativity, essentially, and looking by HIV status and viral status on the right. So here we're looking at the HIV negatives in green, the suppressed in yellow, and the viremic in purple. And you can see no major difference there. When you look by CD4 count, you can see there's some suggestion that the clearance may be slightly different in those with lower CD4 counts. But one of the odd things that Alex and his team do is they measure CD4 counts in all their participants, the HIV negatives as well. And so this graph is actually showing you data from all participants, regardless of whether they are HIV positive or not. So some suggestion maybe there's some difference by CD4 count, but not clear. This is a second study by the NICD called their COVID shedding study that again enrolled hospitalized cases and then followed up with sampling at regular intervals. And here they're looking at the whole study population. And on the left here is the HIV negatives versus positives. And you don't see any clear difference in the time to clearance here by HIV status. When you look by viral load and CD4, you can see the numbers start to get small here. There's some suggestion here that with the viral loads above 400, again, there's a delayed clearance, but this was not statistically significant, probably because of the low numbers in the analysis. However, when they looked at a subset of their study population who had a CT value less than 30 at enrollment, so probably infectious at enrollment, here they could see a difference in the duration of shedding, both by viral load status and by CD4. So on the left, you're looking at CD4, and this red line with the longer clearance is the CD4s under 200. Similarly, on the right, the red line is the HIV viral load over 400, and again showing a longer time to virus clearance. The third study is the lovely study from Cheryl Cohen's group at the NICD, the first C study, which builds on her influenza work. And here this is, oh, sorry, bad echo. Here this is looking at cases in the community, so people are enrolled at households and then sampled frequently again in the community. And here you're seeing on the top by viral load status and on the bottom by CD4 status. And I'm just highlighting here again that for the viral loads above 400 and the CD4s less than 200, you can get a sense that the mean duration of virus shedding, of PCR positivity, is different from the other groups. And when you look at the range, you can also start to see that there are some individuals who have very prolonged duration of shedding. Here you're looking at 5 months or so, the upper limit of the range. So again, some suggestion that there's a difference in this group, either those with very low CD4 counts or with viremia, but again, limited by relatively small numbers. And then this last study is from the next speaker, from Kogi and her team at KRISP, who enrolled predominantly outpatient cases, so cases that were diagnosed at primary healthcare clinics, and again followed these people up regularly with regular sampling, and were able to look predominantly at the difference by HIV status. And here you're just seeing the CT values at the different time points, in red for the HIV positives and in black for the HIV negatives. And you can get a sense from this graph that you start to see beyond the kind of first month, you're seeing still these low CT values, so high viral loads, persisting in the HIV positive cases. So a suggestion that there's these individuals who continue to shed at high viral loads for a prolonged period of time. So to summarize, we have evidence from kind of 4 independent South African data sets that there may be something different in the virus dynamics in people with high viral loads or low CD4 counts with HIV. All of these studies are limited by relatively small numbers of participants with low CD4s and high viral loads. And at some stage, there may be value in trying to pool data from these studies. If there are any keen statisticians in the audience who are looking for a nice project, it might be something to consider. Although some of these studies set out to look at TB, there's much more of a challenge, obviously, with how rare TB is, and so the interactions with TB infection and TB disease have not been well characterized in these studies. And so defining a kind of precise population at risk of persistent infection is not really possible from these data yet. So what about the evidence that in some cases you can actually get a chronic infection that leads to intrahost virus evolution? Here I'm going to show you just 2 reports that we've been involved with from here in South Africa, where we could document the chronic infection and the virus evolution. So the first case some of you may be familiar with was a case from KZN, again from the Commit KZN cohort study. This was a lady in her 30s who was on first-line antiretroviral therapy, but like we're familiar with, had multiple challenges with adherence and with retention in care. She'd been cycling in and out of HIV care for several years. And when she presented to hospital with COVID, she had a very low CD4 count in single figures and detectable virus. She was hospitalized with COVID and required oxygen and steroids, but was discharged after about 9 days, so relatively moderate severity of illness, and then remained largely asymptomatic throughout further follow-up. For various reasons, her viremia and virological failure was not properly picked up and not properly acted upon by the medical team and by the study team. And she was actually left on her failing antiretroviral therapy for some time. And what you're seeing on the right is a graph of, in orange, her SARS-CoV-2 cycle threshold value. So here you're seeing on a scale from low cycle threshold, so high viral load at the top, and you see this persistent SARS-CoV-2 virus for over 6 months. And then in the blue, what you're looking at is the HIV viral load on a log scale. And as I said, there was this long delay, and she was only switched to a new regimen here at this time point. And then you can see her viral load, her HIV viral load suppresses very quickly within a few weeks. And then following that, she now becomes undetectable in her SARS-CoV-2 PCR. So because we had these samples from these time points, we could then go back and sequence these samples to see what was happening to the virus over this time period. And here I'm showing you the phylogenetic analysis. So here's where we compare the genomes from the 7 time points in this individual patient and compare them to all the other viruses that were circulating in South Africa. And what you're seeing on the left is essentially that all these viruses from this single patient were on the same branch of the phylogenetic tree, or the same branch of the family tree. And so we could have confidence that this was the same virus infection that was persisting over time and was evolving over these several months. And that's as opposed to this person being reinfected multiple times with different viruses. When we looked at these genomes in more detail, we could see this complex pattern of virus evolution. And we don't have time to go into the full details of this, but here what we're showing you is some of the different mutations in the spike protein. And as you go up, you're looking at the different time points over the course of this infection. And so what you're seeing is the dynamics here, that you can see some mutations. For example, the one we're very familiar with, the E484K, you can see it appear quite early on in this infection, but then later it then disappears again. So it's taken over by different variants with a different combination of mutations. So there's this kind of dynamic shift in the virus population during a chronic infection. And what we can see when we look into this is that most of these mutations that are emerging over time are associated with escape from different classes of neutralizing antibodies. So this makes sense, yeah, that in a chronic infection, you don't have the adequate immune response to clear the virus infection. But that doesn't mean there's no immune response. There's an inadequate immune response, and that is driving the virus evolution. So it's driving the process why the virus is continually trying to escape the immune response that's elicited. So that was all very well. That was one case, and it seemed that, okay, this happens, but it may be extremely rare because you've only documented one case. So a few months ago, the team at Stellenbosch and Tygerberg Hospital managed to identify another case of chronic infection by slightly different means. So here they had a young HIV positive female actually infected at birth, so vertical transmission, and again was on first-line therapy, but it had multiple challenges with her antiretroviral adherence. She was admitted to hospital in September with complications of her advanced HIV. And she had very low CD4 count and high viral load again at that time. And she was tested for SARS-CoV-2 on admission as a routine and was found to be positive with a high CT. And on investigation, it was discovered that she had previously tested positive back in January 2021. And because of the systems that we'd set up for the genomic surveillance, she had actually tested positive in KZN, in rural KZN. And the lab at Nkosi Albert had stored the specimens and were able to retrieve the specimen from January 2021. So again, we had multiple specimens. We had the January 2021 specimen, and then the specimen on admission, and a further positive PCR. She was still positive a month after admission. And again, we could look at this in phylogenetic analysis and show that these were all on the same branch of the phylogenetic tree. And here we could show that the original infection was with the beta variant, which makes sense in KZN in January 2021. We know that the beta variant was then dominant in the second wave. And what we could show was that, again, multiple mutations emerged over this 7-8 month period between these different sample time points. So again, this was somebody that had remained largely asymptomatic all this time, but we could show clearly that there was evidence of chronic infection with virus evolution. Here's just the kind of genome maps from the two time points. So here showing up in January, the classical mutations from the beta variant. And then by September, you can start to see all these additional mutations, particularly in the spike protein in the receptor binding domain, and also in the S2 region. And for those that are following all these mutations very, very closely, you might start to see some mutations that look familiar from our recent foe, the Omicron variant. So again, these mutations seem to be largely mutations that were associated with escape from neutralizing antibodies. So just to summarize here, we've got two well-characterized cases of chronic infection with very low CD4 counts. Both these cases are notable for predominantly asymptomatic course, and this of course highlights the challenges for identifying these chronic infections. They may not come to light in routine medical care because these people are not presenting unwell, and unless we're sampling for some reason, that they're admitted to hospital for another reason, or they're in a research study, or they're in some surveillance study, then we're not going to detect these chronic infections. It's important that in both these cases, the second case also, SARS-CoV-2 clearance followed soon after introduction of effective ART and adherence support measures. And in both these cases, the mutations that emerged were predominantly associated with antibody escape and or kind of binding affinity to the ACE2 receptor. And even just in these two cases, we started to see some of the same mutations emerging, even though these were different infecting lineages. So the first case was a kind of B.1 lineage from the first wave. The second case was a beta variant. But we could see in both of these that there were similar mutations emerging over time. So one of the questions that we're now challenged with is, we can see these, we can document these cases in people with HIV. There's multiple cases documented around the world in other immunocompromised individuals. And one of the key questions is whether this is one of the mechanisms whereby variants and variants of concern may emerge. So here is just illustrating again this kind of simplified phylogenetic tree, just reminding you that all the variants of concern and variants of interest have all arisen independently. They've all evolved independently and not from each other. And so that has raised kind of three main hypotheses for how these variants might emerge and how they originate. And these are the three theories on the right here. So the first is that these evolve just through the normal process of transmission from person to person, but you don't notice them because they're evolving in some genomic surveillance blind spot. So some part of the world where we're not testing well or we're not surveying well with genomics. The second is what we've just talked about, this chronic human infection with intrahost evolution. And the third is this process whereby the virus may travel between humans and other animals and then evolve in that animal reservoir and then spill over back to humans. So various theories around the types of animals, rats and deer, whereby this could happen. But I think the evidence that we're seeing and the evidence where we follow these chronic infections certainly suggests that this process of chronic infection in humans is a plausible mechanism for the emergence of these variants. A preprint that just recently came out a couple of weeks ago from the team in the UK, where they've collated some of the data from the different chronic infections, and they've looked at the kind of pattern of mutations that are emerging. So I said in our two cases there were some similarities between the mutations. Here they've looked at a much bigger number of chronic infections, and they can again show that there's some common mutations that recur in these chronic infections, and some of these mutations are what we have seen in the different variants of concern. And again, they highlight that most of these mutations are associated in some way with either binding to the host receptor, immune escape, or some mechanism around cell entry. So, taking all this into account, I would argue that we have enough evidence, although none of this is conclusive, around the role of these chronic infections in the emergence of variants. We have enough evidence that we need to take this seriously, and we need to think about this in our longer term public health response. So what does this mean? It means that we may need more monitoring and surveillance for these chronic infections. We need to get a better sense of how frequently these chronic infections may occur in people with advanced HIV, and we need to understand them better. But what's clear is it highlights again the intervention in a sense is very easy, because we know that if we have everyone virologically suppressed for their HIV, then the risk of chronic infection will probably disappear, or will certainly diminish significantly. So it reminds us of this need to plug all the gaps in the HIV care cascade to make sure as many people as possible are virally suppressed. We still haven't properly integrated COVID vaccination into HIV services, and that's something that we really need to do to make sure that these high-risk groups are fully vaccinated, that they get all the doses that they require to get an adequate response to vaccination. And then there's clearly outstanding questions in terms of the research we need to do to understand these chronic infections. We need to understand better the mechanisms of virus evolution. We need to understand whether these chronic infections may still occur in people that have been vaccinated or in people that are experiencing reinfections. And we need to start to understand what's the role of therapeutic agents in managing these chronic infections if we do detect them. So do the antivirals or do monoclonal antibodies have a role in these chronic infections? So that's all I want to say, and hopefully I've convinced you that this is an important topic, and thanks again for putting the symposium on so that we can discuss this. This is just acknowledgement to all the different teams that have generated these data that I've presented to you, including all the network and the national consortium teams, and of course the funders that support this work. And that's just the list of the references that I've mentioned during the talk. So thanks very much, and I look forward to some discussion.
Thank you very much, Dr. Lessells, for such a comprehensive presentation, and also for the simplifying those case studies for us, that give us good detail and a clear picture. Even though the data is limited, I think they give us a clear picture of what's happening in terms of virus evolution. I'll open up now for questions. We've got about 7 minutes for any quick questions and comments. Maybe I'll just start off with one. I'm intrigued by the emergence of mutations during chronic infection and when the viral load also goes up. I'm just wondering what happens before that period. Is it an effect of CD4 T-cell immune responses? What's controlling the virus before the chronic phase?
I think that's a very good question, and I think that's where we do need more work to understand exactly. I mean, clearly one of the questions that we have is, although we know that these chronic infections happen in different types of immunocompromised individuals, they are quite different. So we have seen these cases in people that have predominantly kind of B cell or defects in their humoral immune side, and people with defects in the T cell side, so people with like T cell leukemias and lymphomas and things. I guess if we think, is there something unusual about HIV? Well, we know HIV affects both humoral and cellular immunity. And what's striking about the HIV cases is the fact that largely they are asymptomatic through the course of the chronic infection. Whereas many of the other cases from around the world, they're often in people with hematological malignancies or organ transplants. They're older, they're sick with their COVID, and many of them die. And so there may be something different around the people with advanced HIV in the clinical profile, and that may be telling us something about the immune response. So there may not be enough of a T cell response to clear the virus, but also not enough of a response to make them very unwell and to make them symptomatic. And then there's also not enough of an antibody response. But there's some antibody response that's then driving the evolutionary process. I think one thing we don't know and that we need to understand more is whether escape from T-cell immunity could also partly be driving this process. And that's important because that would be much more of a concern thinking forward if there's the potential for variants to emerge that have more T-cell escape. Then that's obviously more of a concern in terms of our vaccines and the vaccine protection. So definitely still more to learn.
Sure, yeah. Do you have any questions, comments? Amina, you can go ahead.
Thank you so much for just summarizing all that information, as Nobu said. Just taking off from what you said just now. So in the situation of HIV, where we say there's immunosuppression and a poor antibody and humoral and T-cell response, do we think then that viral evolution in this instance is due to defects in viral replication? Because there's no immune pressure. So I was wondering, the issue is, as you said, is there immune pressure and that drives the evolution of variants? Or is it that in an unchecked environment where you don't have a humoral or a T-cell response, that the virus then is free to replicate? And in that free or freer environment, that there's defective replication, and the defective replication then leads to the variants. Do you think that that is the more plausible hypothesis then in this case, as opposed to anything that may happen in a person that has an intact immune response. So.
Yeah, I'm not sure. I mean, I think it's not that there's no immune response. I mean, I think there is an immune response, and that is what is driving the evolution then. But it's clearly insufficient to control the replication. So in that first case, you could see that the CT value was pretty low throughout. So this was virus that was replicating quite well, and I think a lot of that study that I just touched on, where they've looked at the kind of recurrent mutations across all these chronic infections, they're seeing kind of mutations that may enhance the replication over time. So I mean, this virus is clearly trying to survive within the host, but what allows it to survive in the host, what allows it to replicate and continue to enter new cells and things, that may be slightly different from what helps it in the population to spread from person to person. So there may be lots of this happening, where variants emerge that have that if they spill over into the population, they're not going to be successful. They're not going to be successfully transmitted from person to person. I think we don't have that sense of how common it is as a whole, and can just very, very occasionally these variants emerge that also are successful then at spreading from person to person, and far too complex for me to understand. And the immunology is difficult. I think about it from a very simplistic perspective as a clinician, that there's some immune response that can't clear the virus, but the virus there's enough that the virus has to evolve to escape that immune response.
Thank you very much. Seems like our time is up. So it looks like certainly we need in-depth but large sample size studies to understand some of these questions.
Which is difficult. Yeah, it's difficult to do those bigger studies. And with these, of course, we saw that the intervention, if you get these people on treatment, then they clear the virus. And so that first case was an unfortunate case where that intervention was delayed, and that's what allowed us to demonstrate this, unfortunately. Yeah.
Okay. Thank you. Sorry, Beth. It's time for the next talk. Thank you.
Thank you so much. Thanks, Richard and Nobu. So yes, it's time for our next session. And so I'm going to hand over to Marion Loveday to chair from our side. Thanks.
Thank you, Beth. So as Beth said, I'm Marion, and my job today is to introduce COGI to you all. COGI, I'm sure, is known by most of you. For those who've worked in TB and HIV, COGI is a well-known South African and global researcher. COGI has an MBCHB as well as a PhD, and is Deputy Director and Head of the Treatment Research Program at KRISP. COGI is also an Honorary Associate Professor at UKZN in the College of Health Sciences, and she supervised over 30 postdoctoral, doctoral and master's candidates. COGI's contribution to both TB and HIV research and TB and HIV integration spans almost 3 decades, and she's been lead investigator on a number of KAPRISA's clinical trials aimed at optimizing treatment strategies for co-infected patients. She also serves as the site investigator on several international multi-center collaborative clinical trials. And COGI's authored over 110 peer-reviewed publications, as well as a number of chapters in global health books. Kogi also serves on 2 WHO committees, the HIV Treatment Guidelines Committee, as well as the TB HIV Technical Working Group. Kogi also won the quite prestigious Union Scientific Prize in 2013, which is awarded by the International Union Against TB and Lung Disease. So Kogi, thank you for joining us today and being available to join us. And Kogi is going to be talking on the interactions between COVID, HIV and TB and the effects on disease progression. So Kogi, over to you.
Thank you very much, Marion, for that very generous introduction. And thank you to the SAMRC for this invitation to present in this panel. I'm offering a clinician's perspective on the interaction of COVID with HIV and TB. I'm going to be sharing my slides. And just let me know if you're able to see it. Sorry. All right. Please let me know if you can see my slides, Marion.
Not yet, Kogi. Waiting in anticipation.
Okay.
I think you'll have to stop share and then pick this screen with the PowerPoint, Kogi.
Right. Let's go there. Okay. I'm going to put my video off, hopefully. Okay. All right. So it's not giving. All right. Richard, is my slides now visible?
Yeah, we've got them, Kogi.
All right. All right. So is my slides on full screen or is it on presenter view? I'm just trying to ascertain that.
I'm not quite sure, to be honest.
Full screen, I think, Kogi. I see it on full screen. It looks
okay
on my side. Hopefully it's like that with everybody else too.
Okay,
yes,
great. Okay, great. Thank you. So I'm going to share with you the magnitude of the TB, HIV, COVID-19 disease burden. And I'm going to answer a few questions that we all need an answer to. The first is whether HIV and TB is associated with an increased risk of severe disease and death from COVID-19. The second is, does COVID-19 co-infection alter pulmonary tuberculosis clinical manifestation and disease progression? Do people living with HIV or TB have impaired vaccine effectiveness? And a few closing comments on the impact of COVID-19 on the HIV and TB public health response. Sub-Saharan Africa faces the colliding syndemics of TB, HIV, and COVID-19. We know globally there is about 1.5 million new cases of HIV infection each year, with approximately 700,000 deaths, with South Africa holding 15% of this incident TB burden, holding about 230,000 new HIV cases and about 83,000 deaths each year. 10 million new TB cases are notified each year, and as of 2021, with approximately 1.5 million deaths. South Africa holds just over 300,000 new TB case notifications per annum, accounting for about 3.3% of the global burden and about 61,000 deaths. When I last looked at the numbers, globally, there were about 418 million cases of COVID-19 and about 6 million deaths, with South Africa holding about 3.6 million cases and close to 100,000 COVID-19 deaths. So to go to the first question around whether HIV and TB infection and disease is associated with increased risk of more severe COVID-19 disease and death. So I'm going to go through a few publications that will help us answer the question. The very first one is an early paper published in 2020 that came out of the Western Cape by Andrew Bull and Associates. And this was a population cohort study looking at adults that attended government facilities in the Western Cape. Data was collected during our first wave between March and June 2020, and in a catchment population of about 3.5 million people, there were about 22,000 cases diagnosed with COVID-19 and 625 deaths. This study found that with respect to HIV-associated COVID-19 mortality, there was a adjusted hazard ratio of 2.14. So a hazard ratio of 2 compared to someone who is HIV negative. The risk of mortality, they found, was similar, irrespective of viremia, of a viral load greater than 1,000 copies per mole, or a CD4 count under 200. So CD4 and viral load did not influence mortality in this study. They found the current tuberculosis conferred a hazard ratio of 2.7 for mortality and previous TB of 1.5, and that the mortality among sensitive and resistant TB was actually very similar. Interestingly, all deaths occurred during the intensive phase of TB treatment, and most of the COVID-19 deaths occurred among older people. So at the bottom end of this particular figure, you could see the adjusted hazard ratio for COVID mortality. They list a range of risk factors for death. And you can see earlier or higher up on the figure that increasing age plays a critically important role as a risk factor for mortality. And then, of course, the comorbidities of diabetes and poorly controlled diabetes. The lower end of the figure focuses on tuberculosis and HIV, and you could see there that while not as dramatic as increasing age, TB and HIV definitely played a role in increasing risk for mortality. Now to turn our lens on experiences from elsewhere. And this is a paper that was published also early on in the pandemic from data from the United Kingdom. And this used the routine public health database. So it was a population-based research study, and it involved retrospective review of the database where COVID death was compared in HIV-infected and uninfected individuals. You could see in the figure, the Kaplan-Meier shows in the red death from no HIV, which is far lower compared to the line in blue, which is mortality from HIV itself. And they found a higher risk of COVID death in people living with HIV after adjusting for age and sex. So this was the true effect. So we've seen from the data in the Western Cape that age is a strong modifier of risk. But here, despite adjusting for age and sex, they found a hazard ratio of 2.9 for risk for death in HIV. What they also found was that there was a larger association risk, HIV risk and COVID mortality among individuals that were Black versus non-Black ethnicity, where Black individuals in this cohort had a hazard ratio for mortality of 4.3 versus 1.84 in the other cohort. They found there was no conclusive findings on other comorbidities in the population with HIV infection being associated with mortality. They found no association with antiretroviral therapy use, the presence of previous AIDS-defining illness, or viral suppression and CD4 count on mortality risk. So 2 papers so far showing that HIV does increase COVID death, and there doesn't seem to be effect on viral suppression or CD4 count, ART use, or previous AIDS-defining illness. Though the number of HIV positive individuals in this particular study was relatively small, the authors do emphasize that they had sufficient power in this analysis to come up with statistically significant findings, and so these findings hold for this population. A study done in the US, in New York City specifically, assessed outcomes amongst 3,000 known people living with HIV and COVID co-infection, and they compared outcomes to a matched cohort of HIV negative COVID infected controls. So it's a matched case control study. And what they found is that compared to those that were HIV uninfected, patients that had HIV and TB co-infection had similar rates of COVID-19 diagnosis. So what they're saying is that being HIV positive did not confer increased risk of acquiring COVID-19. So the rates of positivity were similar in HIV infection and uninfection. There was a higher per population hospitalization rate amongst HIV COVID-19 co-infected patients, right? So there were higher rates of more severe disease presenting, warranting hospitalization. But what they found was no difference in the in-hospital mortality rates among the co-infected versus the mono-infected patients. They also demonstrated an increasing risk of mortality, of hospitalization based on the HIV disease stage, where compared to stage 1, WHO stage 1 HIV disease stage, the relative risk of hospitalization was 1.29 in stage 2 and 1.69 in stage 3. So increasing risk, and that's your little diagram on the side that's showing your increasing risk depending on the stage. So they concluded that people living with HIV had increased risk of poor outcomes due to higher rates of severe disease that required hospitalization, and that the risk of hospitalization increased with HIV disease progression. This very important paper published from scientists in South Africa, Wasila Jassett and others, showed that HIV was an independent risk factor for in-hospital mortality. So this national cohort evaluated just over 200,000 admitted patients nationally, and 23% of patients in this cohort had died. This cohort the data was collected over a year between March 2020 and March 2021. And what the scientists found was that increasing age was the strongest predictor of mortality. HIV infection increased the risk of mortality, current and past TB again, and that patients that had underlying non-communicable diseases, and this is well established, your hypertension, diabetes, cardiac or renal disease, or malignancy that's diagnosed within 5 years of COVID-19 infection, all were predictors of mortality. Treatment in the public health sector or in KwaZulu-Natal, interestingly, also conferred increased risk for mortality. This study, similar to the ones that we've seen previously, also found that people living with HIV not on ART versus those on ART were more likely to die, right? So there was insufficient data in the UK cohort to make this conclusion, and there was, in fact, this finding was not seen in the small cohort from the Western Cape, but those not on ART were more likely to die in this cohort. And irrespective of whether the patients were HIV positive or HIV negative, what the study found was as you increase the number of comorbidities, there was an increased risk of in-hospital mortality from COVID-19. So all your underlying non-communicable diseases play a role in mortality risk, whether or not you are HIV infected. So to continue with the same study, they went into greater detail to describe ART status, CD4 count above and below 200, and viral load above or below 1,000 as risk factors for in-hospital mortality. And what you could see in this table, the top table, is that patients that were HIV positive, not on ART, were at far higher risk of in-hospital mortality compared to HIV negative patients. That HIV positive patients with CD4 counts less than 200 were at far higher risk of mortality compared to HIV negatives. And those with viral loads greater than or equal to 1,000 copies per mole were again at far higher risk of mortality. They then went on to look at the number of comorbidities and whether that conferred increased risk. And again, they found that those with increasing number of comorbidities had an increase in the risk of mortality. So people living with HIV with comorbidities and the number of comorbidities conferred higher risk. And so this paper emphasizes what Richard has already mentioned in his presentation, that individuals at high risk for mortality would benefit from vaccine prioritization, as well as early referral for treatment and care. So when we know our patients fall into these categories, low CD4 count, high viral load, underlying comorbidities, and they now newly diagnosed COVID-19, they require close clinical observation. Moving on to a larger study, which is called the WHO Global Clinical Cohort, that assessed findings from 24 countries in a cohort of people living with HIV that were admitted with COVID-19. And this study, this retrospective evaluation, assessed in-hospital mortality and clinical severity of disease. They defined severe or critical cases as those with a oxygen saturation of less than 90%, a respiratory rate of more than 30 beats per minute, received ECMO, they had ICU admission, or they had received an inotrope or a vasopressor. Patients that had oxygen saturation of more than 90 or a respiratory rate of less than 30 were regarded as mild or moderate disease. Overall, again, about 23% of patients died, and the mean duration of from hospital admission to death was about 9.5 days. The figure that you see is basically describing at the outcomes of patients as in association with their presenting illness at hospital admission. And what you can see is that those that presented with severe or critical illness at hospital admission, right? You had about 60% of patients that were discharged alive. Mortality, however, was about 35%. So more than 1 in 3 patients actually had a very bad outcome. And then the dark black piece on the diagram is those that were still hospitalized at the time of data close. So these were your outcomes amongst patients that are presenting really ill that have COVID-19 and HIV co-infection. Again, what they found is HIV was an independent risk factor associated with in-hospital mortality, and that, like the JASID paper, that age above 65, being a male, and the presence of diabetes and hypertension were additional risk factors for mortality among people living with HIV. This paper concluded that HIV remains an independent risk factor for severe or critical illness at hospital admission and for in-hospital mortality. Richard presented this paper by Alex Sigal's group, and he looked at the viral evolution in this cohort. And so I'm turning my lens on understanding the impact of HIV viremia and the influence of variants on what we actually observe. So what this was an observational cohort, and there were 93 patients that had both HIV and COVID-19, with 30% having previous TB and 10% with current TB. And what they found was that the disease severity and the immunologic changes differed in the first and the second wave. During the first wave, there was no difference in the level of severity and the clinical presentation amongst patients that were HIV infected and uninfected. They did, however, observe some modulation in the COVID immune response in the first wave. In the second wave, with a new variant, there was a worsened disease severity. There was increased need for supplemental oxygen use. There was lower CD4 counts during the co-infection with COVID-19 and a higher neutrophil lymphocyte ratio. And what they found is as soon as the COVID cleared, there was a rebound in the CD4 count. And wave 1 in the dark bar and wave 2 in the gray bar in the figure below with A and B, showing the viremia, the COVID, the HIV viremia that seemed to be different between the two waves. They also did some immunologic studies, and what they found is that there was reduced neutralization of the SARS-CoV-2 infection amongst those that were immunocompromised, right? So there's less ability for the body to clear the virus. And Richard discussed the emergence of the variants, and they found that there's a synergistic relationship between HIV infection and COVID-19 that can alter the clinical outcomes of COVID infection. So I'm now turning our attention to tuberculosis. Does COVID-19 co-infection with TB alter clinical manifestation and disease progression of TB? So this paper was published very early on in the pandemic from a group from northern Italy, from Lombardy, where they attempted to characterize COVID-19 in patients with active tuberculosis. And what they've had is 20 TB patients that were diagnosed with COVID within a month of being diagnosed with TB. What they found was about 2/3 cleared their COVID infection within 2 weeks. And one of their patients died. And when they looked at their chest radiographs and the impact of COVID-19 on TB lesions, they found that in 12 patients, the lesions improved. There was no change in the one patient that had pulmonary TB, and that in 7 patients, there was worsening of the TB on chest radiograph. And when they looked at the COVID lesions on the chest radiograph. There were 4 that developed new onset pneumonia, 3 that had mild to moderate interstitial disease, and 1 that had a ground glass appearance. So clearly there's an influence of COVID on TB and on and the COVID itself, being able to discern the changes on chest radiograph that these authors describe. In this,
sorry to interrupt you. You've got 6 minutes left.
Okay, all right, thank you. So when we looked at COVID-19 and TB in the study done in the Philippines, what they looked at a matched cohort of just over 500 patients, with 100 of them with confirmed previous or current TB. They found that those with TB compared to those without TB had a 2-fold higher risk of mortality, 25% lower recovery, a shorter time to death, and a longer time to recovery. So again, emphasizing the interaction between COVID and TB. This study done in Southeast Asia also looked at the susceptibility to COVID that TB patients had to COVID and to COVID disease severity. A very small case control study of 36 patients and 13 diagnosed with TB infection based on IGRA positivity. And what they found is that patients with the co-infection with TB had a greater likelihood of severe symptoms, greater rapidity of symptom onset, and a greater likelihood of complications. Paper from Kayalicha looked at rifampicin-resistant TB patients, and interestingly, only 30% of patients received a COVID test, despite patients presenting with respiratory symptoms. And among these, there were just 18 that tested positive. These were all hospitalized patients. And strikingly, 61% of these rifampicin-resistant COVID-19 co-infected patients died. There was no significant difference between those that died and survived. And the mortality amongst the COVID-negative patients was 6.6%. And the authors conclude that mortality was significantly higher amongst the RIF-resistant patients who also tested positive for COVID-19. It is likely that the sicker patients were offered the COVID-19 testing, and they suggest a possible pathological biologic interaction between COVID and RIF-resistant TB, leading to this excess mortality that was observed. Is vaccine effective among people living with HIV? And when we look at the J&J viral vector vaccine, 500,000 healthcare workers in the Sisonke trial, they found similar neutralization antibody response in healthcare workers that received the J&J vaccine that were HIV positive and well controlled. And it was similar to the HIV negative participants. And this was irrespective of whether or not they had past COVID infection. There was, however, a reduced neutralization antibody response in HIV-infected individuals that were non-vaccinated. And the reduction in the ability of neutralization was observed most in those HIV viremic individuals. So HIV viremia plays a role in vaccine uptake, and the ability to neutralize, and no difference, it seems, in the capacity to neutralize in people living with HIV versus HIV uninfected. Small studies looking at the Pfizer and Moderna vaccines, the RNA vaccines, you could see 14 people living with HIV that received the Pfizer vaccine and only 9 with the Moderna vaccine, and they assessed antibody titers a month post-vaccination. After the first dose, the titers were fairly, it was widespread over a range of 0.4 to more than 250 units per ml. But after the second dose, all titers were high at more than 250. So there was a good anti-RBD antibody response after vaccination. and with 2 doses, and despite patients being HIV positive, there was no rebound or change in HIV virologic control despite vaccination. When we look at the Astrozenica vaccine, again, they looked at patients that were stable on ART for more than 3 months and viral loads that were suppressed, and found no difference in the antibody response in HIV positive and negative people at days 28 and 42 post-vaccination. And also, they report, one of the only studies that reported adverse reactions, and they found no difference in people living with HIV and those without. The Sinopharm inactivated vaccine, small numbers of individuals, and they found it to be safe and immunogenic, and developing comparable antibody response. In our Sisonke study, evaluating the J&J effectiveness amongst HIV-infected healthcare workers in South Africa, this is a preprint that assessed HIV-infected in the blue bars and uninfected in the red bars, and you can see vaccine effectiveness was slightly reduced amongst HIV-infected individuals for hospitalization and those requiring critical care or ICU. The vaccine effectiveness was maintained at about 65% against death amongst people living with HIV that received the vaccine. The impact of COVID-19 on HIV and the public health response, we know that we're going to be seeing over the next 5 years substantial impact of the pandemics and the lockdown on high incidence settings such as ours. And modeling data predict more than 6 million excess TB deaths by 2025 as a result of decreased diagnosis, treatment initiation, and treatment completion that occurred as a result of the pandemic. Likewise, an increased mortality in HIV by 40%, likely as a result of a 6-month disruption in ART service delivery. Of course, we may also observe a surge in HIV drug resistance, and this is a concern as there may be a potential impact on managing TB and HIV and undermining the gains made over the last decade. The work done by Jenshi and Nigel in CAPRISA during the lockdown, where they assessed the impact on facilities in eThekwini and in northern KZN, and they found that the lockdown reduced attendance to health facility by 57%. It reduced HIV testing by 47%, ART initiation by 46%, but ART pickups for patients in chronic care remained unaltered. Pre-COVID, we saw a substantial reduction in TB mortality from 2014 to 2018. But you could see the red bars that HIV deaths remained unchanged since 2014. And we know that TB is the commonest cause of death. Post-COVID-19, or not really post-COVID-19, but a recent publication showed that TB mortality increased by 100,000 deaths in 2021 alone. So clearly there's a reversal in TB mortality as a result of the pandemic. So in summary, HIV, we can see from the literature that I showed you, is a significant independent risk factor for severe or critical illness at hospital admission, as well as for in-hospital mortality. That the risk of hospitalization increases with HIV disease stage among those not on antiretroviral therapy, those with CD4 counts less than 200, and those that are viremic. Increasing comorbidity and increasing age is also risk factor for hospitalization and poor outcomes amongst people living with HIV. The paper by Alex Segal showed that changes in the virus makes some variants alter the disease course and the effect on people living with HIV compared to other variants. Preliminary data from low burden countries have shown that COVID-19 in TB is more severe, with higher mortality and more complications. And of course, with RIF-resistant TB, the data from South Africa showing really high mortality in co-infection with COVID-19 and RIF-resistant TB. We found that vaccine effectiveness is maintained against death and slightly reduced vaccine effectiveness against hospitalization and in-hospital critical care requiring requirements among those that have HIV compared to HIV uninfected. Thank you very much. And just like to thank all our funders, and back to you, Chair.
Kogi, thank you so much for that very comprehensive and interesting presentation. We've run out of time, so I'm going to, we can all save any questions we've got for the final discussion, if you don't mind. But I'm going to pass straight back to Beth now so we can get on with Cobus's presentation.
Thanks, Marion, and thanks, Kogi. And so now we're handing over to Terusha to chair our last session, and to Cobus. Thanks.
Thanks so much, Beth. Morning, everyone. I'm Terusha Chesi, and I'm based at the MRC HIDRU offices in Durban. I've known Cobus Herbst since 2007. Dr. Cobus Herbst is a medical doctor. He has a specialist degree in public health and a master's in bioengineering. He is also the director of the DSI MRC South African Population Research Infrastructure Network, also known as SAPRIN. And he's a faculty member and the director of population science at the Africa Health Research Institution, as well as the PI on the Population Intervention Program. In 2013, Cobus established the in-depth data repository containing longitudinal demographic and course-specific mortality data on over 3 million individuals from 25 demographic surveillance sites in lower and middle income countries. And this work has also been extended in a collaborative project with researchers from the London School of Hygiene and Tropical Medicine to establish a repository of longitudinal population-based HIV serosurveillance data from 10 sites in Africa, and this is known as the Alpha Network. So good morning to Cobus, welcome and thank you for your talk today, and Cobus will be discussing the interaction between COVID-19, HIV and TB and the effect on health services delivery.
Thank you, Teresa, for the kind introduction. I just want to confirm that you can see my slides.
Yes, I can see your slides. Thanks, Cobus.
Thank you. I'm going to talk about the interactions between COVID-19, HIV and TB and the effect on health service delivery. Kogi already characterized the COVID-19, HIV and TB pandemics as syndemics. Richard Walton already argued in a Lancet paper more than a year ago that COVID-19 is not a pandemic, but a syndemic. This notion of a syndemic was first conceived by Merrill Singer, a medical anthropologist, in the early 1990s. When considering the interaction between COVID-19, HIV, and TB and the effect on health service delivery, this notion provides a useful reminder of the complexity of interacting co-present or sequential diseases, as is the case with not only COVID-19, HIV, and TB, but notably also including NCDs. Each is syndemic in its own right. Additionally, there are social and environmental factors that promote and enhance the negative effects of disease interaction, something that was starkly illustrated by the convulsion of looting and unrest that swept through KwaZulu-Natal and Gauteng during the third COVID wave in July 2021. This approach reconfigures conventional historical understanding of diseases as distinct entities in nature, separate from other diseases and independent of the social context in which they are found. Richard and Kogi in their presentations have already made the case of how these diseases interact at biological and individual level to influence viral evolution and disease progression. In my presentation, I will examine how these interactions impact on health service delivery and access and subsequent health outcomes. Kessel recently published in a preprint a systematic review of the literature on the impact of COVID-19 pandemic and anti-pandemic measures on tuberculosis and HIV and AIDS and a couple of other diseases. These figures document the types of impact they identified from the literature. On ART retention and engagement and care, including changes in ART consultations, initiations, risk of ART interruption, and viral loads. Amongst HIV testing and diagnosis, testing utilization decreases, number of new cases and positivity rates, and other preventions, including impacts on voluntary male medical circumcision and PrEP usage. For tuberculosis, in terms of clinical outcomes, including rates of successful treatment completion, loss to follow-up, treatment failure and initiation, diagnostic tests, notification of cases and incidents, service provision, including inpatient and outpatient care, reallocation of staffing and diagnostic infrastructure, and shortage of medical supplies, and case reporting and surveillance. In other words, the current literature have documented a wide-ranging impact of the COVID-19 syndemic on HIV and TB-related services. Specifically, in the case of tuberculosis, Kogi has already mentioned some of the modeling results, but the modelers predicted, due to disruption of service, an 11% excess cases over 5 years and 16% excess deaths over 5 years, with reduced case detection resulting in a 25% drop on average of case detection and 13% excess deaths over 5 years. In terms of observed impacts, range from an 87 to 20% decline in notifications, with treatment success completion rates showing a range from 70% to no decline. In terms of HIV, modeling results here due to ART interruption, I mentioned estimate excess cases between 1 and 16% higher over 1 year, and excess deaths between 39 and 87% higher over 1 year. A reduced viral suppression could cause a 15% increase in excess cases over 1 year and 18% increase in excess deaths over 1 year. The observed impacts show a mixed impact on ART consultations, moderate decrease in viral load and CD4 tests, and an all-over decrease in testing. This figure is interesting in showing the response by patients in various surveys to their experience with ART. So proportions of patients reporting difficulties with ART refill, unable to get ART refill, decreased ability to adhere to ART, and risk of ART interruption. In a Global Fund snapshot, examines the disruption patterns caused by COVID-19, as well as the adaptive measures that the Global Fund supported programs have taken to respond to these challenges. It's based primarily upon data collected and aggregated through programmatic spot checks, recording information from April to September 2019 and comparing that to data collected from April to September in 2020 across 502 health facilities in 32 countries. On this slide gives a breakdown of reasons for changes in patient attendance from April to September 2020, seeing that the reasons for these changes are reported to be more patients being redirected from and to other facilities, scope of specific services reduced, large numbers of patients presenting with respiratory infection symptoms, communication failures about reactivation of any servers that were previously suspended or reduced, etc. In terms at individual level, fear, mistrust, uncertainty of getting infected with COVID-19 from facility visits, disruption in public transport, lockdown or stay at home that restrict movement, delayed care seeking, and other changes. HIV referrals and HIV testing showed an initial steep decline with a gradual recovery over time. Drug-sensitive TB diagnosis and screening, as well as screening or testing for HIV and TB patients, also showed a steep decline immediately after the start of the epidemic, with a steep decline and a subsequent recovery again. The following service adaptations were reported from this survey. In terms of the management of health workers and community health workers, staff were reassigned to different units in the facility to deal with these impacts, or were temporarily transferred to different facilities over time, new staff recruitment, volunteer recruitment, and part-time staff increasing their hours. In terms of adapted measures for delivery of health services, facilities extended drug prescriptions, so for a longer period, to ensure patients had long-term and uninterruptible access to medication, prioritization of high-risk patients, integration of services during a single visit, redirection to alternate facilities, increase in home-based care, changing in location, e-health or telemedicine to reduce the need for actual visits, and different ways of delivering prescriptions, as we see with the CCMDD program here in South Africa. If we look closer to home, Genshi from CAPRISA showed steep declines in HIV test, ART initiations, ART collection visits, and an increase in missed visits and programmatic data from 65 clinics in KZN. The team at ARI and the Slabisa subdistrict, using a different data source by research assistants collecting data at the clinics during the lockdown period, did not see this sharp decline in ART visits in local clinics. Body and an editorial in the SAMJ first drew attention to the steep decline in TB testing, and in a figure from a paper by Jogan Pillay compared tests done over a longer period between January 2020 and February 21 compared with the expected number of tests, also showing a significant drop in tests at the start of lockdown with recovery later in the period. In Umkanyakudi, Northern KZN, Khan and Gareta in unpublished data shows a similar decline in TB testing, but over a slightly longer period shows a recovery in test numbers associated with a decreased yield over the last 6 months. Pulmonary TB cases identified more recently have a lower bacillary burden than earlier periods, and the change in patient demographics, male-female ratio, suggests that current increase in TB screening with ultra, whilst welcome, may be missing high-risk populations such as men. If we look more broadly at health outcomes, over the last few years, we have noticed a decline in HIV incidence and a shift to older age groups, particularly in men. It will be important to assess whether the shock to the HIV prevention and care system by COVID-19 pandemic had any effects in the short and longer term on these trends. This graph from the MRC's report on weekly deaths in South Africa is well known. It will be important to understand the exact source of these exact deaths, although it's likely that at least a substantial proportion is due to unnotified COVID-19 deaths. Some of the excess may be attributed to other causes exacerbated by the impact of COVID-19 on health service delivery or individual healthcare seeking behavior. In conclusion then, although a range of impacts have been described on TB and HIV services, many of the impacts have shown to reduce over time, and more research is needed to understand the nature of this resilience to gain insight into improving health service resilience in the case of future shocks. However, as shown by Richard, even relatively small numbers of people living with HIV and AIDS not linked to ART or difficulties with retention and care could potentially impact on the COVID-19 pandemic. And Covet has shown how uncontrolled HIV disease impact on COVID-19 outcomes. Most of the published research focused on the immediate impact of the COVID-19 pandemic on health services. Research is needed to understand longer-term impacts and policy changes that might be required to improve health systems resilience. For example, it appears that TB diagnostic service demand has returned to pre-pandemic levels, but with differences in positivity rates and patient demographics, hinting at longer-term effects. Fully teasing out the ongoing impact of COVID-19 pandemic on the health system will require enhanced ability to do record linkage across different health information systems, as evidenced by the exemplary work that have originated from the Western Cape Health Data Warehouse Group. Thank you, Terusha. Back to you.
Thank you very much, Cobus, for a very comprehensive presentation. Just while we're waiting for questions from the audience, we have 10 minutes for discussion. Can you describe some of the work that you're doing in the in-depth and alpha network and how that was adapted for data on the COVID-19 pandemic, and how you see data systems evolving to mitigate the risk of these excess deaths?
Yeah, I can't speak about in-depth and alpha network because these are almost at this point in time historical networks, but I can definitely talk about what happened in SAPRIN, which is DSI funded harmonization and combination of the 3 established HDSs in South Africa, namely Agincourt in Mpumalanga, Dimamo in Limpopo, ARI here in KZN, and then a new one that is currently busy with their baseline survey in Gauteng, including Hillbrow, Atteridgeville, and Malusi, and then a new one in Cape Town that will be starting up in a couple of months' time, and 2 new ones, one actually here in Durban, in eThekwini, and another rural site in East London. What we've done in the COVID outbreak, when fieldwork was stopped due to COVID lockdown, we could switch our data collection to telephonic data collection, which is part of our protocol, and introduce then symptomatic screening and questions around the impact of non-pharmaceutical measures on households and individuals, questions on mental health, as well as later on vaccine willingness and hesitancy. So that is an ongoing piece of work that is ongoing in SAPRIN, and we regularly analyze those data and share that on policy forums. Thank you. In terms of the excess deaths, I think these surveillance sites with the verbal autopsies that they do there can provide valuable insight into the cause-specific component of those excess deaths. And there's a project that I'm involved in with Steve Tolman at Agincourt that will be looking at exactly this in 15 sites in Africa and Southeast Asia.
Thanks so much for this. I'm just going to check the chats to see if there are any questions from the audience. can't see any at the moment. I think another question from me is just, based on the data that Richard was presenting and Kobe was presenting, do you have any data around chronic COVID infections or long COVID and what that means for HIV? Is that collected in the SAPRIN network?
We've talked about that, but the problem is the difficulty in diagnosing that, especially at population level. So I don't have any specific information about it. I saw Amina had her hand up.
Thanks so much. Amina?
Thanks, Kobus, and thanks so much for that presentation. I just wondered, with all the data that you presented and that other people also have about effect on services, there really needs to be a concerted effort to strengthen services. And just from the work that you've done, have you or are you testing any models within each of the nodes that could be implemented either within an NHI context or outside an NHI context in the current context, just to see how services can get back on track? I think there are two parts to this. One is the provision of services, or maybe three parts: the provision of services, then the actual health facility and staff, and then people's uptake of services. So anything that or can think of, or any models that we can use, because we have a lot to catch up with as we move forward.
Yes, I think there are two pieces of work in SAPRIN nodes. One is the work on elderly at Agincourt that provides including also, in fact, the first study of NICD and Cheryl Cohen that is nested in that site, that provides an excellent basis for us to look at service impacts in such a population base. And then at ARI, you may be familiar with work of Mariam Shamanesh and peer support. And I think that there's also an opportunity there to look at innovative models to enhance care and counteract the negative longer-term impact on health service delivery.
Thanks, Amina. Thanks, Clovis. We have about 4 minutes for the interview discussion. I don't see any other hands. Do you have any? Oh, sorry, there is a question from Nita.
Hi, thank you very much for the excellent presentation, Clovis. So based on your data and even that was presented by Prof. Lessells, how has the Province Department of Health responded to the issues around service delivery using this data set, both short-term in terms of enhancing HIV, TB awareness with COVID work, and then of course the second part of it is vaccine uptake or other intervention uptake in terms of prevention and treatment of TB and HIV specifically, and of course STIs. Thank you.
I want to hand over to Richard I think he can answer it better. The bit that I want to answer is about vaccine hesitancy and willingness. For example, in the local district health services, we've been sharing those insights in terms of reasons why people give, in terms of why they would not really opt or is not willing to be vaccinated. And the local health services have changed their messaging as a result of that. And so we've also, by we I mean SAPRIN, have presented this vaccine on several occasions at what is called the National Policy Data Observatory, which is a forum of directors generals. And I know that that information has been shared at higher policy levels with regards to a vaccine messaging. Richard?
Yeah, thanks, Cobus. I'm not sure I have any real insight into what the provincial health department have done. I mean, I think in terms of the vaccination, I think most people probably realize it's been quite centrally controlled at a national level, like many things, and they've been quite slow to take up recommendations about integration of COVID vaccination into other health services, so particularly antenatal care, as well as HIV care. And I think that's something that really needs to be addressed going forward. Yeah, but I'm not sure. More generally, I think they recognize these issues, but I mean, I guess for me, what's always striking when we look at these data is actually how resilient the system is, and how hugely effective ART is, even if there are gaps and there are challenges and things. And to me, it always highlights that we need to simplify service delivery to the vast majority of people who do well with ART and don't need much in the way of medical care. If we can further simplify that, then we should, and so that our limited resources are really focused on those that really need the care and support. But that's of course easy in principle, but not easy in actually delivering it. Over.
Thank you.
You're muted, Teresa.
Oh, thanks, Richard. There is an additional question in the chat, but it's just gone 10 to 11. I'm going to just check with Beth and Amina if we continue with the discussion, or we can hand over to you to close the session and close the symposium.
Maybe we can just take the quick question in the chat bar.
Although we have looked at the impact of lockdown on hospital admissions at the Slabisa Hospital, we didn't find any direct evidence of the impact of reduced admissions due to alcohol-related trauma.
Thanks so much, Cobus, and thanks for a great session. I'm going to hand over back to Amina now to close the symposium.
Thanks so much, everybody. And I think thanks especially to Richard, Kogi, and Cobus for sharing your insights. Before I close and just give a summary, I wanted to ask each of you, what your high-level message would be, or if you have anything else to say on the topic that you presented, given the questions and the discussion. So I'll just go around if there is anything. Richard, from your side.
I mean, I think we do need to take these interactions seriously, and we do need to think about them now, thinking more about the long-term response to this pandemic. I think for the last 2 years, there's clearly been a lot of discussion around people living with HIV and COVID. I think although Kogi showed the data that they're at increased risk of morbidity and mortality, that was clearly relatively modest compared to the effective age, and so from that perspective, there's never been that we've not put that strong an argument forward that they are a group that needs specific attention. But I think when we put everything together, like you've done in this symposium, they clearly are a group that requires additional attention, and for these different reasons. And I think even though the science has been really incredible in 2 years, we've never done science like this, there's still a lot of gaps in our understanding of things to guide the kind of longer term response. So I think there's still room for lots of work to get a better understanding and to really think long term. And I should have said at the end of my talk, it's not just us that are thinking about this. I mean, internationally, there's more focus on this now. There was another piece in JAMA just last week from Larry Corey and other prominent experts kind of saying that we really need to pay more attention to this. So I think it is getting increased recognition, and we need to lead that science again here in South Africa, I think. Yeah, thanks.
Thanks, Richard. Kogi, from your side?
Hi, Amina. So even though age and cumulative number of comorbidities are strong risk factors, we have the largest population of people living with HIV in our country, and so the scale of when we look at the increased risk of poor outcomes among people living with HIV and the scale of the epidemic of HIV in our country, we have a big problem in our hands. And so therefore there's two things from that. The first is the importance of education awareness around vaccination and prevention among people living with HIV, and increasing the uptake and prioritizing vaccination in that population. And secondly is about strengthening HIV viral control, but making sure that patients at high risk for poor outcomes should they get infected with COVID is minimized, and enhancing viral suppression, rapid switching to more robust regimen in patients that are failing, and just a general all-round strengthening of the HIV program, I think will go a long way in mitigating the impact of COVID in our population. Thank you very much.
Thanks, Kogi. Cobus, any last words from your side?
Yeah, I want to use the opportunity to re-emphasize the last point in my conclusion in terms of integration of health information. From my experience trying to replicate the Western Cape data warehouse here in KZN, it's really extraordinarily difficult to get access to the different data sources such as tier.net, HPRS. Although some of the COVID data sources like DATCOV and the NICD notifications are more readily available, but having access to identifiable information so that these data sets can be integrated and we can really look at these health systems impacts that we have talked about, and I think be able to make valuable policy contributions as a result of that analysis for us, especially research groups that have access to large research populations. It is really something that I think should be addressed.
Yeah, thanks very much, Cobus and Kogi and Richard, for just sharing those ideas. I want to just say thank you to everybody. I'm just going to put up a final few slides as we close. Just tell me when you can see them.
Yep, you can see them.
It's coming. So thank you so much to everyone. So we really wanted to look at the interactions between COVID-19, HIV, and TB, specifically on the effects of viral evolution, disease progression, and health service delivery. And a big thank you to Richard, Kogi, and Cobus. You know, you brought together a lot of literature, each of you from a different angle, and we've managed to put that together in today's symposium. Thanks also to the three chairs and to Beth for her overall coordination of the symposium. I think, there were several key points that have come through from Richard's presentation, looking at the tendency for high HIV viral load and low CD4 cell count to delay SARS-CoV-2 clearance, the observation that most recurrent mutations in immunocompromised people are associated with ACE2 affinity, immunoscape or viral packaging optimization, the idea that it's the asymptomatic infection that is usually associated with viral persistence and evolution. And there are several questions and hypotheses about how variants arise. Then from Kogi's presentation, the association between early TB or early stage of disease, HIV infection, not on ART, and we're seeing similar signals in children as well. The association between people living with HIV and low CD4 cell counts or high viral loads associated with in-hospital mortality. And these come from several studies, and that's compounded by age and chronic illness such as diabetes and hypertension. I think the other alarming factor is the estimated increase in COVID death, not COVID deaths, but TB deaths by 2025. So an excess of 6 million TB deaths that we should document by 2025, and a 40% increase in HIV mortality by 2025, just because of what's gone on in the last 2 years. So I think, and then Cobus, of course, highlighting that this is a syndemic with lots of effects on the health system. So I think what we've really said is, we need, and I think we've said these in several fora, enhanced monitoring and surveillance, strengthening HIV and TB care. And Richard, you made the point about simplifying service delivery so that those people who are in need of more advanced care can receive that care. Then to vaccinate priority groups and advocate for vaccination in these priority groups. I think we need to pay attention to staff resilience. And then to pivot to prioritizing high-risk patients using other models or testing models such as home-based care. The gaps that we need to address are around mechanisms of viral evolution, risks of chronic infection post-vaccination and in reinfections, the question about therapeutic agents in chronic infection or in people living with HIV or TB. And the key question is how to strengthen services or health systems. And I think the main point also that came through is that we need a long-term response to this endemic. That in South Africa, we particularly have to pay attention to people living with HIV, and that we've got to pay attention to the integration of health information. So thank you very much, everybody, for all your participation today. A special thanks to our speakers and the chairs. And we look forward to taking this discussion forward through various other fora that some of us belong to, the Variants Forum or other groups that we belong to. And thank you everyone for all your contributions. And Ari, I see your hand up, so over to you. Do you have an announcement?
Yeah, just in the chat for Amina.
Oh, okay, thank you, Ari. So we'll post the recording on the website. And if you don't get it, then please email Ari in 2 days' time so that he will send you the link. And then I think with permission of the speakers, we can obviously share the presentations, although they will be on the website with permission of the speakers in PDF format. So thank you very, very much, everybody, and have a good week further. Bye.
Thank you.
Thanks.
Thank you.
Bye.
Bye, everyone.
Thank you. Thank you very much. Bye.
Thank you very much.
Thank you.
Thanks,
Bye.
everyone. Bye.
Vele mina ulishiya lingaphandle kulo mfana. Hawu, wake wakhumbula ukuthi kune-meeting uthi uyongena kuyo ufica abantu sebewo-living. Vele ibe u-9 to 11 i-symposium.